morning, students. In my earlier classes, I have already discussed about some of the biological macromolecules which are present in the cell. Those molecules are either protein, carbohydrates and so on. Today, I will discuss on another biological macromolecules which are present in the cell called nucleic acids. Nucleic acids are the molecules that store information for cellular growth and reproduction. If we broadly classify the entire nucleic acids, it can be divided distinctly into two major groups. One is called deoxyribonucleic acid or DNA and ribonucleic acid or the RNA. These are the polymers consisting of long chains of monomers called the nucleotide. So, what is nucleotide? A nucleotide consists of a nitrogenous base, a pentose sugar and a phosphate group. That means, in other words, we can tell that in a particular nucleotide, we have a nitrogenous base, one sugar moieties and one phosphate group is there. Now, this is the structure of a nucleotide. Say this is a pentose sugar. In carbohydrate classes, I have already discussed that what is pentose, what is hexose. So, this is a pentose sugar. If we number this carbon, then this is the first number of carbon, second, third, fourth and fifth. So, this is a pentose sugar and the first carbon is being occupied by a nitrogenous base. So, here one nitrogen is attached to these sugar moieties and to this fifth carbon of this particular sugar one phosphate group is attached, and when this sugar, nitrogen and phosphate groups are there, then we can call it as nucleotide. Now, what is this nitrogenous bases which are there along with this particular nucleotide molecules? So, if we see this nitrogenous bases in nucleotide, it consists of two general type. One is the purine, another is the pyrimidine. Now, if we see the structure of purine and pyrimidine, then we can find that this pyrimidine is a nitro containing heterocyclic molecule. Now, here if we see further this pyrimidine, it has got three main derivatives. One is called cytosine, it is symbolized as C, thymine T and uracil U. When to this heterocyclic nitro containing ring one imidazole group is being attached, it forms the purine. And if we see the derivatives of this purine, we can find that adenine and guanine. That adenine is symbolized as A and guanine is symbolized as G. These are the derivatives of purine. Now, if we further go into these derivatives of purine and pyrimidine, we can find that this adenine and guanine, this A and G, they are the purine derivatives are present, both are present in RNA as well as DNA molecules. Whereas, if we see the pyrimidines and pyrimidines derivatives, then we can find that this cytosine is present both in DNA and RNA, whereas thymine is present only in DNA and uracil is present only in RNA. That means structurally, if we see the structure of these pyrimidine derivatives, that is thymine and uracil, we can find that say rest of the structures are same, both the molecules except this methyl group which is attached to these thymine molecules. And these thymine molecules, when this methyl group is removed or this methyl group is not attached, we can call it as uracil. That means, in other word, we can tell that this uracil, methyl containing uracil is the thymine, or demethylated thymine is the uracil. So, these are the structural differences, and here this thymine is present in DNA, and in RNA thymine is substituted with uracil, that is U. That means, in short, what we can conclude from this nitrogenous bases, we can tell that A, G, C is common, that means adenine, guanine, cytosine is common in both DNA as well as RNA molecules. Whereas, in case of DNA, when we are considering DNA, then we can tell A, G, C and T, whereas in case of RNA, when we are talking about RNA, we can write A, G, C and U. This U, this thymine is changed with uracil. This is the difference between one DNA molecule and one RNA molecule. So, these are all about the nitrogenous bases, and these nitrogenous bases are attached to the first carbon of the pentose sugar. So, when we are coming to this sugar moieties, now as I have told you that 5 carbon sugar moieties, and that is why we are calling it as pentose sugar. Now, this sugar, this pentose sugar are also of 2 types in case of DNA as well as RNA. In case of RNA, this is the simple ribose sugar. This is the ribose sugar. See, this is 1, 2, 3, 4 and 5 carbon sugar, and this is the ribose sugar in case of RNA molecule. But here in case of DNA, it is called deoxyribose. That means one oxygen is missing in the sugar moieties, 5 carbon sugar. So, what is that? See, in the second carbon, here you see one H and OH is there, but here in this deoxyribose, you see oxygen is missing. No oxygen is bonded to this carbon atom, and that is the reason why it is called deoxyribose, and which is present in the DNA molecule. The sugars have their carbon atoms numbered with the prime to distinguish them from the nitrogenous bases, and to this particular carbon atom number 1, here the nitrogenous bases are being attached, and this is the major difference with the ribose sugar, 5 carbon sugar with DNA and RNA. Now, coming to this nucleoside and nucleotide. A nucleoside consists of a nitrogenous base linked by a glycosidic bond to carbon atom number 1 of the ribose or deoxyribose. So, irrespective of the ribose sugar, to the first carbon these nitrogenous bases are attached to both in DNA as well as RNA molecules. Nucleosides are named by changing the nitrogen base ending to ocene for purine and idine for pyrimidines. The nucleotide is a nucleoside that form a phosphate ester with the carbon atom number 5 hydroxyl group of ribose or deoxyribose. Nucleotide are named using the name of the nucleoside followed by 5 prime monophosphate. So, if we see the actual structure, then we can find that when we talk about this nucleoside, nucleoside means ribose sugar, and to this first carbon this nitrogen base is attached. So, here you see this is the adenine group is attached to this, and here if it is the deoxyribose, then this O will be missing, HH, then it is the adenosine group is there. In case of nucleotide, say nitrogen and sugar, this is nucleoside. When we are talking about nucleotide, then I have already shown you that nitrogen, sugar, and to that one phosphate is attached. See the structure. This is nitrogen, sugar, to this phosphate moieties are attached to this. So, it is called adenosine 5 prime monophosphate. That means, it is the adenosine 5 prime monophosphate, that means to the 5 carbon atom this phosphate group is attached. So, monophosphate, why? Only one phosphate group is attached. So, when we are naming this nucleoside and nucleotide, then how we are naming? In case of RNA, I have already told you that here the bases are, this nitrogenous bases are A, G, C and U. So, see A, G, C and U, this nitrogenous 4 nitrogenous bases are there. In case of nucleoside, we are calling this adenine, guanine, cytosine and uracil as adenosine, guanosine, cytidine and uridine. So, these are the nucleoside naming. When we are talking about the nucleotide, then adenosine 5 prime monophosphate, as I have already mentioned that here nucleotide are named using the name of the nucleoside followed by 5 prime monophosphate. So, similarly adenosine 5 prime monophosphate, guanosine 5 prime monophosphate, cytidine 5 prime monophosphate, uridine 5 prime monophosphate and so on. In case of this DNA, here it is deoxyribose. So, obviously deoxyadenosine, deoxyguanosine, deoxycytidine and so on. When we are naming the nucleotide, then deoxyadenosine 5 prime monophosphate or DAMP, deoxyguanosine 5 prime monophosphate, deoxycytidine 5 prime monophosphate, deoxythymidine 5 prime monophosphate. So, these are the names based on the type of molecules we are discussing. So, these are the nucleotide and nucleoside and differences between this nucleotide and nucleoside. Now, when we are talking about this monophosphate, diphosphate, triphosphate and so on, then this additional phosphate group can be added to the nucleoside 5 prime monophosphate to form diphosphate, triphosphates and so on. What does it mean? See, this is a nucleotide, nitrogenous base, sugar and one phosphate is there. If we want to attach another phosphate group to this, that means here if we want to add upon another phosphate group, then it can be attached, and here it forms the adenosine diphosphate. To this diphosphate, when we are interested to incorporate another phosphate group to this, then it forms the adenosine triphosphate or ATP molecule. And as we all know, we have already learned that ATP is the major energy source for all cellular activities. So, these ATP molecules are playing a significant role in different metabolic activities which are performed by the cell. Now, coming to this primary structure of the nucleic acid. The primary structure of the nucleic acid is the nucleotide sequence. The nucleotides in nucleic acids are joined by phosphodiester bonds. The 3' hydroxyl group of the sugar in one nucleotide form an ester bond to the phosphate group on the 5' carbon of the sugar of the next nucleotide. What does it mean? It is, say, this is one nucleotide, this is another nucleotide. So, just till now what we were discussing, that what is nucleoside and what is nucleotide. We have learned nucleoside is sugar plus nitrogen base, and when one phosphate is being attached to this, it forms the nucleotide. That means, in case of nucleoside, no phosphate is attached. In case of nucleotide, nitrogenous base, sugar moieties plus phosphate. That means, sugar plus nitrogen base is the nucleoside, sugar, nitrogenous base and phosphate is the nucleotide. Now, when one such nucleotide is being attached to another nucleotide, then this ladder, this linkage or this chain like formation is taking place. Now, how this chain is being formed? Now, here you see this is the carbon 1, 2, 3, 4, 5. First carbon is already occupied by this nitrogenous base, and third carbon, in the third carbon this phosphate group of another nucleotide is going and it is getting bonded with the phosphodiester bond. Now, when this type of linkages are taking place, then one nucleotide is getting attached to the another nucleotide, and this phosphate, when it is coming to the next nucleotide, the third carbon sugar moieties of the another nucleotide, then it is getting linked, and in this way the chain is formed, and this chain is called the primary structure of the nucleic acid. I think if I show you in little elaborate way, say this is the sugar moieties, this is the third carbon, this is the fifth carbon of the sugar, and the first carbon, it is the nitrogenous base. This is the fifth carbon. To this fifth carbon phosphate group is attached. This phosphate is once again getting joined to the third carbon of the second nucleotide. Here nitrogenous base is there, fifth carbon, here phosphate is there, and this phosphate is being attached to the third nucleotide and sugar moieties and third carbon of the third nucleotide, and here from this phosphate is being attached to the fourth nucleotide molecule, the sugar molecule, the third carbon of this fourth number of this nucleotide, and in this way this 1, 2, 3, 4, and that way nucleotides are being linked up and they form a chain like structure. These nitrogenous bases may be A, T, G, C, A, U, G, C. So, say here A, C, G, T, these are the nitrogenous bases. So, a nucleic acid polymer has a free 5' phosphate group at one end and a free 3' OH group at the other end. The sequence is read from the free 5' end using the letter of these bases. Say for example, 5' A, C, G, T, 3'. That means, if we want to write the structure that how these nitrogenous bases are attached, then we have to write 5' A, C, G, T and then ending with the 3'. So, this is the example of the primary structure of the nucleic acid. You see, this particular linkages has got a very close similarity with the primary structure of the protein molecule. What I have already discussed, that one amino acid is being attached with another amino acid, the second amino acid is getting attached with third, fourth, fifth and so on. What is happening in case of this amino acids? When they form this protein structure, that in one particular amino acid we have already learned that there is an amino terminal and one carboxyl terminal of that particular two groups are there. So, when one amino group is getting attached with the carboxyl group of another amino acid, they form the peptide linkage, and this peptide linkage joins that one amino acid to another, and that way the primary structure of protein is formed. Here the similar thing is happening with this particular primary structure, where you can see that one nucleotide is getting attached with another nucleotide with the third carbon of this pentose sugar, and the phosphate group is being linked up and the chain is getting increased, and that way primary structure of this nucleic acid is formed. Now, here this is the example of this RNA primary structure. Here this T is replaced by this U, and here this 3 to 5' ester linkages are there. So, here if we want to tell, then we can tell this that 5' A, C, G, U and 3'. Whereas, in case of DNA, you see U is replaced with the thymine. So, if we want to tell the structure of this primary structure of this particular nucleic acid, then we can find that 5' A, C, G, T and 3'. So, these are the primary structure of this DNA and the RNA molecules. Now, coming to the secondary structure. So, when we are talking about this primary structure, we have already learned that this is a very simple structure which is there and joining the nucleotide together. In the secondary structure, we can find the DNA double helix. That means, in DNA there are two strands of nucleotides that wind together in a double helix form. The strands run in opposite direction, that is anti-parallel to each other. What does it mean? That means, if one strand is from 5' to 3', another strand will be from 3' to 5'. So, this oppositely that numbered, that 5 prime, if it is starting, another end will be 3 prime, and when these two strands are coming, that means they are anti-parallel to each other. When they come and they bind each other and form this type of structure, it is called the helix like structure, and this is the secondary structure of the DNA molecule. The bases are arranged in a step-like manner, and the base pairs are held together by hydrogen bonding. The pairing of the bases from the two strands is very specific. The complementary base pairs are AT and GC. That means one purine is getting linked with another pyrimidine. That is the linkage. No two purine or two pyrimidines are there. One purine is getting linked with another pyrimidine. So, A is getting bonded with T, G is getting bonded with C. So, two hydrogen bonds are formed between A and T, and 3 hydrogen bonds are formed between G and C. Each pair consists of purine and pyrimidine, so they are the same width, keeping the two strands at equal distance from each other. So, this is the adenine and this is the thymine, one is purine, another pyrimidine. So, two bonding, hydrogen bonding is there, that means A double bond T and G triple bond C. See here G guanine, that 1, 2, 3 hydrogen bonding and cytosine. So, these are the helix structure, double helical structure of this DNA. ATGC linkages, base pairs are there, and which are associating the two strands in a organized manner. DNA double helix model is first introduced by Watson and Crick in 1956, and they got Nobel Prize for that. The bases are oriented perpendicular to the helix axis. They are hydrophobic in the direction perpendicular to the plane of the bases, cannot form the hydrogen bond with water. The most common DNA structure in solution is the B form of DNA. Under conditions of applied force or twists in the DNA or under low hydration conditions, it can adopt several helical conformation which are referred to as A form of DNA or Z DNA and so on. See, here is one nucleotide strand, another, and you see AT, GC, GC, AT, and that way this helix structure is there. It is formed in between and they are associating the two strands together. While coming to this A form, B form and Z form. So, B form is the normal DNA molecules, and under extreme environmental or extreme conditions we can get A form of DNA or Z form of DNA. When we are comparing the properties of this type of DNAs, if we consider the B form as the standard, we can find that this A form it is the right handed, B form is right handed helix, Z form is left handed helix, and there are very many differences between this base pair, this helix diameter, rise per base pairs, base pair per helical turn, helix pitch, etc. are different in each form of DNA. Now, if we see at a glance, we can find that the A form of DNA is very thick and short, B if we are considering as normal, then Z form is thin and long. So, these are some of these DNA helix. The DNA double helix is formed by two right-handed complementary strand of nucleotide coil around each other. Right-handed is the term used to describe the symmetry of the three-dimensional object. It means that if you place the thumb of your right hand along the long axis of the molecule and curl your fingers in the chain twists in the same direction as your finger. Now, if suppose this is the double helical structure, and now if we wind this double helix structure to the thumb, and if it is forming this right-handed helix, it will be wound like this, and this is called the right-handed helix of DNA. And in case of left-handed, this double helical structure is winds and it forms, and in this way it winds up with the central axis, and this is the left-handed helix of DNA. So, if we see this other properties, the storage of DNA in eukaryotic cells, mainly the animal, plant and fungi, DNA is stored in the nucleus, which is separated from the rest of the cell by the cell membrane, and within that the cell sap is called nucleoplasm. That is, that I have already discussed those things, that inside the nucleus, in case of eukaryotes, that nucleus has got its own nuclear membrane, and that is separating this nucleus from the other organelles, and nucleus has got the bi-bi layer, that nucleus has got its own this is the one nuclear envelope which is separating the nucleoplasm from this cytoplasmic fluid. And here inside this the nucleic acid or the chromosomes are there. DNA is organized into chromosome during cell division. During replication, the DNA is stored in a compact ball-like structure called the chromatin and is wrapped around the protein, which is the histone protein, to form nucleosomes. So, these are some of these things. So, inside this nucleus, the replication of DNA is taking place. When one eukaryotic cell divides, the process is that the process they adopt is the mitotic cell division. The cell, in this particular cell division, the cell split into two identical daughter cells. The DNA must be replicated so that each daughter cell has the same copy number of this nucleic chromosome. DNA replication involves in several processes. First is the DNA must be unwound and separating the two strands. The single strands then act as a template for synthesis of the new strand, which are complementary in sequence. The bases are added one at a time until the two new strands are formed that exactly duplicate the original DNA which are produced. The process is called semi-conservative type of DNA replication, because one strand of each daughter's DNA comes from the parent DNA and one strand is synthesized, which is new strand. During this process, the energy which is required comes from the hydrolysis of the phosphate group as the phosphodiester bond form between the bases, as we have already learned that thing. Now, if we are talking about this semi-conservative DNA replication, then this is the parent strand, and first what it is doing, it gets unwound. Now, one is the 3 prime to 5 prime, another is 5 prime to 3 prime. So, anti-parallel to each other. So, when these two strands are getting unwound, getting separated, then new strand, this old strand is the template strand for synthesis of this new strand, and this way these new strands are synthesized. And when these new strands are synthesized, it has got one old, the template strand, parental strand, and new synthesized strand, and these are called the semi-conservative type of DNA replication. The enzyme helicase unwinds the several sections of the parent DNA. At each open DNA section called the replication fork. So, here this is the open up form, that is the replication fork of DNA polymerase, catalyzes the formation of 5 prime to 3 prime ester bond of the leading strand. So, here this is the parental strand got opened. So, one end is the 3 prime end, another is the 5 prime end. This is one parental strand, this is another parental strand. Now, there is a leading strand and another is the lagging strand. So, when 3 prime to this 3 prime end is there, so 5 prime to 3 prime, this leading strand is starts synthesizing and they form the complementary base pair of this particular new strand. Whereas, in the lagging strand, which grow from 3 prime to 5 prime direction, is synthesized a small short section of this nucleotide base pair, and that is called the Okazaki fragment. This Okazaki fragment, with the help of the enzyme DNA ligase, they join and to form a single 3 prime to 5 prime strand, and in this way the replication process is taking place in DNA. That means replication means DNA to DNA. So, from one parental strain, the template is that is used as a template and new synthesized strand is taking place. Coming to this ribonucleic acid or the RNA. RNA is the most abundant than DNA. There are several important differences between RNA and DNA. The pentose sugar in RNA is ribose, and in case of DNA is deoxyribose. In RNA, uracil replaces the base thymidine, that means U pair with A. RNA is single stranded while DNA is the double stranded. RNA molecules are much smaller than the DNA molecules. If we categorize this RNA, then we can differentiate this RNA as ribosomal RNA or rRNA, messenger RNA or mRNA, and transfer RNA or the tRNA. Now, if we see in the RNA molecules, the different types, different compositional variation of these three RNAs, ribosomal RNA or rRNA, of the total RNA present, it constitutes 75%, whereas mRNA is 5 to 10% and tRNA is 10 to 15% of the total RNA which is present. If we see the functions of this particular RNA in the cell, inside the cell, then we can find that rRNA is the major component of the ribosome. mRNA carries the information of protein synthesis from the DNA in the nucleus to the ribosome, and tRNA brings amino acid to the ribosome for protein synthesis. So, this way this RNA, different RNAs are doing a very important role in cell system. ribosomal RNA, when we are talking about, this is the ribosomes are the site for the protein synthesis. They consist of ribosomal RNA 65% and protein 35%. The function of rRNA is to provide a mechanism for decoding mRNA into amino acids, and it interact with the tRNA during translation by providing peptidyl transferase activity. rRNA is the most conserved, and that is the reason why nowadays we rely upon the rRNA data for identification of any unknown species. So, this is the most important part of rRNA. Messenger RNA is the mRNA, carries the genetic code to the ribosome. They are strands of RNA that are complementary to the DNA of the gene for the protein to be synthesized. There are another type of RNA called small interfering RNA, or it is called siRNA. They are the class of double stranded RNA molecules, 20 to 25 nucleotide base pair in length. siRNA is involved in the RNA interference, that is RNAi activities pathway, where it interferes with the expression of a specific gene. They have implicated in the wide range of functions such as cell growth or apoptosis, that is programmed cell death, development of neural plasticity, remodeling, and even insulin secretion. Now, coming to the tRNA. Transfer RNA translate the genetic code from the messenger RNA and brings specific amino acid to the ribosome for protein synthesis. Each amino acid is recognized by one or more specific tRNA. tRNA has a tertiary structure that is L-shaped. One end is attached to the amino acid and the other bind to the messenger RNA by the 3 base complementary sequence. If we see the structure of tRNA, it has got a 5' terminal phosphate group. That this particular tRNA has a CCA tail, that is cytosine adenine sequence at the 3' end of the tRNA molecule. This sequence is important for the recognition of tRNA by enzymes critical in translation. In prokaryotes, the CCA sequence is transcribed in some tRNA sequence. In most prokaryotic tRNA and eukaryotic tRNAs, the CCA sequence is added during processing and therefore does not appear in the tRNA gene. This tRNA has a D arm, that is 4 base pair stem ending in a loop that often contain dihydrouridine. It has got also anticodon arm, that is 5 base pair stem whose loop contains the anticodon, and it has got also a T arm, that is the 5 base pair stem containing the sequence, that is T psi C, where psi is the pseudouridine. So, as I have told you already that it is the tRNA which is L shaped. See, this is the L shaped tRNA. If we say this is the loop where this anticodon loop is there, and if we see the structure that say in the 3' end of this particular hydroxyl group, one CCA group is there. It forms the ester bond to the amino acid, and this is the acceptor stem. and it has also got a 5 prime end, as I have already discussed. That I have also told you that it has got that loops, that one is the D arm, another is the T arm, and another is the anticodon, and this anticodon arm is the 5 base pair stem, and it sends the messenger RNA which is coming with the polysome and forming the nucleotide, and it is sensing the amino acid for this particular that mRNA, and this is the importance of this particular tRNA, and it brings the amino acid to that particular site. When coming to this transcription, that several steps occur during transcription. A section of DNA containing the gene got unwinds. The mRNA is synthesized using complementary base pairing using uracil, that is U, replacing the thymine, that is T. The newly formed mRNA moves out of this nucleus to the ribosome in the cytoplasm, and the DNA rewinds. So, if we see, I have already discussed that DNA replication is taking place in the nucleus. DNA replication is taking place. Transcription, when it takes place, DNA forms the mRNA. When mRNA is formed, mRNA comes out of this nucleus to this outside of the cell, and it is coming to the cytoplasmic fluid of the cell. And these two main processes involved in protein synthesis are the formation of mRNA from this DNA, that is the transcription, and conversion of tRNA to proteins at the ribosome, that is the translation. Transcription takes place in nucleus, while translation takes place in the cytoplasm. So, as soon as mRNA is formed, mRNA is coming out from this nucleus to the cytoplasm, where this translation is taking place. So, here if we see the transcription central dogma of this cell, see here replication is taking place, DNA to DNA is the replication. When DNA is undergoing transcription, it forms DNA, forms this mRNA. When mRNA is formed, this mRNA comes out from this nucleus to the cytoplasmic fluid, where actual protein synthesis is going on, and these amino acids are producing this protein called the translation process, which is going on in the cell with the help of this nucleic acid. Now, coming to this genetic code. The genetic code is composed of nucleotide triplets. In other words, 3 nucleotides of mRNA, that is specify one amino acid in a particular protein. So, what we can tell that 3 nucleotide is a codon which specify for one particular amino acid. One codon is specified with one particular amino acid. The code is non-overlapping, that means there is no overlapping of this particular code. This means that successive triplets are read in order. Each nucleotide is part of only one triplet codon. The genetic code is unambiguous. Each codon specifies a particular amino acid and only one amino acid. For example, the codon ACG codes for the amino acid threonine and only threonine. The genetic code is degenerate. in contrast, each amino acid can be specified by more than one codon. Each code is nearly universal in nature. Almost all organisms in nature, from bacteria to human being, are exactly the same genetic, use exactly the same genetic code. The rare exceptions include some changes in the genetic code of mitochondria and in a few protozoan species. Now, coming to this mRNA codon and associated amino acids. So, I have already told that these genetic codes are the triplet codes. So, if we are taking this first letter, second letter and third letter, this that nitrogenous base pairs, that see here how these codes are formed, that C U C A G, this is first letter, U C A G, and this is the third letter, U C A G. Now, if we form this genetic code, then three letters triplet codes are there, U U U, U U C, U U A, U U G. See, U U U, U U C, U U U, U U A and U U G. So, these are the codes, triplet codes. Now, in reality, we have 4 base pairs and 3 nucleotides form this one code. So, in nature, we have 64 such nucleotides, this codes which are coding this amino acid, but in reality what is happening? In reality, we have only 20 standard amino acid, I have already told, and they form this permutation and combination. With their permutation and combination, these proteins are formed. So, what is happening in reality? If this 64 codons are there and 20 amino acids are there, and if each codons are responsible for coding only one amino acid, then what will happen? There will be 44 such codons, that this codons for which will be there. So, during protein synthesis, when it will be coming with any permutation and combination, and if these 44 codons, if there is no amino acids are there, so any point of time when such codons will be coming, no amino acids will be there for such type of codon, and any time this protein, that chain synthesis will get terminated. But in reality, it is not happening like that. That means, what is there? In reality, for each codon, there are certain amino acids which are there. Say for example, for GGU, GGC, GGA, GGG, for these 4 codon, only one amino acids are there, which is coding glycine. AGU, AGC, AGA, AGG. See, AGU, AGC is serine, AGA, AGG is arginine, and in this way, for this natural, this 64 codons, there are certain amino acids which are coded, and that is the reason why it is called, it is it is degenerative. So, but there are some exceptions, like AUG is the start codon which codes for the methionine, and UAA, UAG, UGA are called the stop codon, and only one codes which only codes the trypsin is the UGG. This is the exception of the genetic code. Now, if we see another example, say we can, let us take any example, CGU, CGC, CGA, CGG. Now, here if we see these 4 codons are coding one amino acid, that is arginine. If we see that, say these first 2 codons are same for this particular amino acid, but the third codon is varying. In case of glycine also, you see first two codons are constant, same for the whereas the third amino acid is varying. That means the third codon is not that important, that third nucleotide base pair is not that important for this particular recognition of this amino acid, but first two are very important, and that is the reason that first two are constant for identifying the particular amino acid, and that is called the Weibull hypothesis of genetic code. Now, if we come to this reading of this genetic code, suppose we want to determine the amino acids coded for this following section, that is 5 prime CCU dash AGC dash GGA dash CCU dash 3 prime, where we know proline is CCU, GGA is glycine, AGC is serine, and CUU is leucine. Now, if we want to write this proline, serine, glycine, leucine, we can write, but when we are writing the actual codon, we have to have the 5 prime CCU, AGC, GGA, CUU and 3 prime. While writing this codon, there should not be any punctuation, any comma, any full stop, any dash or any such type of use of punctuation while writing the genetic code, and this is the correct way of writing the codon. Nowadays, 16S rRNA for prokaryotic and 18S rRNA is used for identification and characterization of unidentified living organisms because they are conserved in nature. RNA being conserved in nature, such identification are considered to be the most reliable. Nucleic acid and nucleus present inside the cell behaves as the brain of the system, as the entire cellular metabolism is controlled by the end product of the central dogma, that is the protein. And with this, today I am concluding my lecture, and I have tried to give you some idea about that how cell is undergoing division, cell division, growth, replication and reproduction is going on with the help of this nucleic acid, it is DNA and RNA molecules. In my next class, I will discuss about another macromolecules which is playing a significant role in cellular activities, and that is the lipid molecules. Thank you very much.